I am representing today the Laboratory for Ornamental Plants and Vegetable Crops of the Bydgoszcz University of Science and Technology. And during this presentation, I would like to tell you a few words about my research, about the projects that we do in our department related to the application of nanoparticles in plant biotechnology, especially in cryobiology studies. So basically, nanoparticles, as you probably know, are very, very small structures of a very tiny diameter of a maximal 100 nanometers. And due to this very small size, nanoparticles have specific, unique properties, physical, chemical properties, which differ from the same particles, which are bigger. So nowadays, nanotechnology and nanoparticles are being utilized in various scientific disciplines such as biology, chemistry, physics, technology, materials, and everything which is between those disciplines. So, for example, also in medicine, in biophysics, in electronics, optics, and so on. And nanotechnology is also being used in agriculture. For example, nanoparticles are being used for the production of fertilizers, for the production of herbicides, fungicides. For, as you know, gold and silver, for example, have some antimicrobial properties. They are also used for the production of nanosensors and also in genetic engineering as carriers of the transgenes used for the genetic modifications process. And currently in our laboratory, we are doing some scientific projects awarded by the Polish National Scientific Center related to the application of nanoparticles in plant biotechnology. And shortly, I would like to tell you a short history about our research in this field. Because basically, in our laboratory, we are using nanoparticles for three main purposes: for the reproduction of plants in vitro, for breeding purposes and the creation of new cultivars, and also we use it in biodiversity protection in cryopreservation studies. So when we started our research, first we wanted to study how different nanoparticles affect the in vitro development of plants. And we observed that depending on the concentration, but also on the type of nanoparticles, we can have a different impact of those materials on the development of plants in vitro. So for example, here you can see that with chrysanthemum, the addition of nanoparticles at high concentrations to the culture medium, well, did not really improve the regeneration efficiency of the plants. Actually, they were even regenerating worse than the control. But for example, in bleeding heart, the addition of gold nanoparticles stimulated very efficiently the multiplication ratio of this plant species. And also, as I said, we did some studies regarding the influence of nanoparticles on the genetic stability of plants, because it is well known that nanoparticles, nanomaterials may have some impact on the genetic stability of the plants. And indeed, we observed that when adding nanoparticles, especially silver nanoparticles, to the culture medium, the adventitious shoots that are being formed in vitro, well, their development is being reduced, decreased, but also we observed that the plants produced in the presence of nanoparticles may show some mutations, and which are manifested in the form of the change of the flower color or its shape. So basically, all of those research, those findings, those results made us think about the possibility of applying nanoparticles in cryopreservation, in the storage of biological material in liquid nitrogen. Because there is very little knowledge on the effect of nanoparticles on the cryopreservation efficiency, especially in plants. Like I said at the beginning, nanoparticles, due to their very small size, have some very unique physical properties. And one of those properties is that they have a very high thermal conductivity, and therefore they may enhance the cooling and rewarming pace, for example, during cryopreservation. And as you all know, this is crucial when establishing a successful cryopreservation protocol. And in some previous studies of other authors, they found that, for example, gold nanoparticles may be used for the PCR reaction, or also zinc nanoparticles can be used in cryopreservation of animal semen, because, like I said, those nanoparticles affect the thermal conductivity and the cooling rate. That's why we thought about using this also in plant cryopreservation. And in our studies, in our research project, we used bleeding heart, also known as Lamprocapnos spectabilis. This plant originates from Asia. It is mostly an ornamental plant due to some exceptional flowers, some very interesting shape. It is a member of the Papaveraceae family. Therefore, it also has some other applications. This plant species can also be used, is used in folk medicine. It is also a source of some valuable alkaloids, which can be used, for example, in the treatment of depression. Also some lactones, which can be used in cancer treatment. But the extract from this plant can also be used in dermatology and cosmetology, because it is known that the extract from this plant slows down the aging process of the skin. And what's more, the extract from this plant can also be used in antiseptic, as an antiseptic agent. And this plant species can also be used as a source of resistance genes. However, very little research has been done so far in biotechnology of this plant species. That's why we are developing some new knowledge related to this species and to various applications of plant tissue culture in bleeding heart. And basically, the aim of the studies that I will tell you now was to verify the usefulness of nanoparticles in the cryopreservation of plant genetic resources. We used different types of nanoparticles, silver, gold, and zinc, which were used at various concentrations during different steps of the cryopreservation procedure. And now I would like to tell you a few words about the things that we did and the results that we obtained, because in the first experiment that we performed, we have tested the usefulness of gold nanoparticles in plant cryopreservation. And for this purpose, we used the encapsulation vitrification protocol, during which the single node explants are first pre-cultured on a medium with an increased sucrose and ABA concentration for 1 week. Then the shoot tips are being isolated, encapsulated, dehydrated with the loading solution in a plant vitrification solution, after which, of course, they are being stored in liquid nitrogen and then recovered in vitro. And the modification of this method that we used was the addition of gold nanoparticles at 3 different concentrations. And these nanoparticles were either added into the pre-culture medium to the encapsulation to the alginate bit matrix or to the recovery medium. And in this experiment, we used nanoparticles with a diameter of 13 nanometers. And besides that, of course, in order to verify the utility of the effectiveness of those nanoparticles, we included 3 controls. One control which included plants that were cryopreserved without the addition of nanoparticles. One control where only the stabilizing medium was added without nanoparticles. This is a medium which is used for the production of nanoparticles, and also a non-cryopreserved standard was used, which was only cultured in vitro. And afterwards, we have subjected our plant material to various analyses. We analyzed, of course, the recovery potential of the plant material, some of its biometrical features, and also studied some cellular events such as the oxidative stress, such as the genetic stability, and some of those studies are already done. Some others will still be performed in the course of future studies, such as the cytogenetic analysis, the histological and ultrastructural studies, and also the ex vitro analysis of the plant material. But so far, we have learned that the addition of nanoparticles, of gold nanoparticles, significantly affected the successfulness of cryopreservation, although the effect of these nanoparticles depended depending whether they were added to the pre-culture medium, to the recovery medium, or to the alginate bit matrix. And we found that when adding gold nanoparticles at low concentrations to the alginate bed, to the capsule, the efficiency of cryopreservation of the recovery potential increased by 20%. And also the plants which were produced from, recovered from the capsules that contained gold nanoparticles had the greatest fresh wave. On the other hand, the addition of nanoparticles to the recovery medium, especially at the highest concentration, had a negative influence on the survival of the plant cells and also on the shoot's fresh wave and the rooting potential and the number of roots produced by the plants. Some other observations that we made demonstrated that plants after cryopreservation produced generally more multiple shoots in comparison to the non-cryopreserved standard. But this is quite a typical situation, a quite typical phenomenon, because as we observed in our earlier research, due to some cryo injury of the apical meristem, the axillary meristem can get activated, and that results in the production of some multiple auxiliary shoots. And here the nanoparticles did not affect this phenomenon. It was basically quite the same as in the control without the nanoparticles. And also the addition of nanoparticles did not affect the elongation of shoots. Because again, we observed that the shoots that were produced after cryopreservation in general were shorter in comparison to the in vitro grown control. That's because of the alginate capsule, which well, inhibits a little bit the growth of the shoot tips, and here also the nanoparticles did not affect those events. We also studied some oxidative stress events in the cells by verifying the activity of 4 different enzymes, and we found that as it was predicted, the addition of nanoparticles did affect the oxidative stress in plants. As you can see, the addition of higher concentrations of nanoparticles into the pre-culture medium or to the recovery medium increased the activity of those enzymes. However, when the nanoparticles were added to the protective capsule, alginate capsule, there was no significant increase in the activity of enzymes. So there was no additional stress, oxidative stress put on those plant tissues, which is important because, like I said earlier, this combination allowed us also to increase the survival rate of the plant material. We also studied the effect of cryopreservation on the metabolic activity of the cells, because like I said, bleeding heart can be a source of some interesting metabolites, and that's why we also wanted to study how do cryopreservation and nanoparticles affect the synthesis of some of the plant metabolites, primary and secondary metabolites. And again, we found that as it is often observed in cryopreservation studies, the use of the plants which were recovered after storage in liquid nitrogen contained significantly less pigments than the standard plants that were not stored in liquid nitrogen. This is quite a normal situation because pigments usually are quite unstable, and due to stress, they get degraded quite easily. So this is what happened here, and the nanoparticles did not affect this. Basically, there were no bigger differences between those events. We also studied the membrane stability index to be able to follow how do the addition of nanoparticles during those different cryopreservation steps affect the integrity of the cell membranes. Because as you know, during cryopreservation, one of the biggest problems is that the formation of ice crystals may disrupt the integrity of the cell membranes and lead to the death of the cell, but also nanoparticles as cytotoxic materials may affect this parameter. And after doing this membrane stability index evaluation, we found that the results of those analyses were consistent with the previous ones related to the oxidative stress analysis and the recovery potential analysis, because the only experimental object where a significant decline in the stability index was observed was when nanoparticles were added at the highest concentration to the recovery medium. And as I said earlier, when we added the gold particles, nanoparticles to the recovery medium, the survival was also significantly lower. So this is why, because those nanoparticles destroyed the cell membranes and caused the cells to die. And one of the most important things which need to be verified when developing a cryopreservation protocol is to study the genetic stability of the plants. So we also did this in our research. And again, the results that we found were quite consistent. So we observed that when the nanoparticles were added to the pre-culture medium, some minor genetic variation was observed, but also when the highest concentration of gold nanoparticles was added to the alginate matrix. However, these were some very small changes, and when we performed the AMOVA analysis, the analysis of molecular variance, it showed that this was actually non-significant. But what is important is that the combination which was most optimal, which guaranteed the highest survival of the plant material, was genetically stable. There were no mutations detected in that experimental object. So those results that we obtained also made us think about the possibility of using also other nanoparticles, because as I said earlier, different nanoparticles might affect the efficiency of in vitro culture of different plant species in a different way. So that's why in the second phase of our research that we are doing currently, we used, in addition to gold nanoparticles, also silver nanoparticles and zinc nanoparticles. But this time we also used different concentration, because as I told you earlier, when we used higher concentrations of nanoparticles, we observed a negative impact on the survival, on the rooting, on the membrane stability integrity index. That's why in the second phase, we used lower concentrations of nanoparticles, 5 and 15 ppms, and we also used a smaller diameter of nanoparticles. Previously, it was 13 nanometers, and now we are using 6 nanometers because, well, we believe that the smaller the nanoparticles are, the greater the chance that they will be absorbed by the cells from the medium, for example, and perhaps they will work more effectively. So these are some initial results that we obtained. And as you can see, the addition of silver nanoparticles and gold nanoparticles into the alginate matrix increased again significantly the survival of the plant material compared to the control where no nanoparticles were used. However, there were also some combinations, some experimental objects, which had a negative impact on the survival of the plants. And these were mainly the experimental objects in which the nanoparticles were added into the pre-culture medium. Because in the second phase of the experiment, we added the nanoparticles either into the pre-culture medium or to the alginate matrix. We didn't use them again in the recovery medium because, as we observed earlier, they had a negative impact on the survival. And also in the second phase, we studied another cultivar, and here again we found that the addition of nanoparticles, especially of silver, gold, but also zinc this time, nanoparticles had a positive influence on the survival of the plant material. It was over 20% higher in comparison to the control. And again, some experimental objects in which the nanoparticles were added into the pre-culture medium had a negative impact on this parameter. So this is quite interesting because when we analyzed further some of the biometrical parameters of the plants, we observed that none of the nanoparticles nor their concentration affected the multiplication of shoots. However, we also found that in contrast to the results related to the survival of plant material, the elongation of shoots which were pre-cultured on the medium with the addition of nanoparticles was in generally better in comparison to the experimental objects when where the nanoparticles were added to the bead matrix, to the synthetic seed. This is quite surprising because previously, in the previous slide, I showed something quite different because I will just remind you that the addition of nanoparticles to the capsule usually increased the survival, But the addition of the nanoparticles to the pre-culture medium stimulated a better elongation of shoots after rewarming, and this was even more evident in the second cultivar studied. Here also, we observed that when the nanoparticles were added to the pre-culture medium, more shoots, a better proliferation of shoots was observed than in all the treatments when the nanoparticles were added to the alginate matrix. And the same again in this cultivar goes when we talk about the elongation of shoots. Again, the longest shoots were obtained from the shoot tips, which were pre-cultured in the presence of nanoparticles. And on the other hand, those experimental combinations in which the nanoparticles were added to the alginate matrix produced significantly shorter shoots, although the survival efficiency in those combinations was much higher. So in order to study and understand this phenomenon in the future studies, we will also analyze again some anti-oxidant mechanisms, and we will also do some microscopic observations using electron microscopy to see what happens exactly to the cells, to the organelles during the cryopreservation process, and how do the nanoparticles affect the structure, the ultrastructure of the cell. But basically, to sum up, these studies shed some new light on the possibility of applying nanoparticles in plant studies, and especially in cryopreservation. We found that the effect of nanoparticles is strongly related with their concentration and also the method of plant treatment, depending if the nanoparticles are added to the culture medium or to the alginate matrix. We also found that silver, gold, and zinc nanoparticles applied at low concentration during the encapsulation step of the cryopreservation protocol have a beneficial effect on the survival and recovery of the shoot tips and bleeding heart without altering the DNA sequence of those plants, although the final effect is cultivar specific, because for example, in one of the cultivars, only gold and silver nanoparticles were the most effective, and in another cultivar, also zinc was effective, even though there was no significant impact of those nanoparticles in the first cultivar. On the other hand, the addition of nanoparticles into the recovery medium after rewarming has unfavorable impact, which results from the disruption of cell membranes and the oxidative stress damage. Our studies also confirmed some minor genotoxic effects of gold nanoparticles if they are used in suboptimal conditions, but this may also suggest that nanoparticles could be perhaps used in mutation breeding as a chemical mutagen when added at a sufficiently high concentration. And some of our future studies will also focus on the utility of other nanocolloids such as copper and titanium in the cryopreservation studies, especially after adding those nanomaterials to the loading solution or to the plant vitrification solution, because this is something that we haven't studied yet. If you are interested in more of our research, you are of course welcome to read some of our articles related to the application of nanoparticles in plant biotechnology. And if you are interested in any cooperation, we are always open and look forward to working with you.